Pharmacokinetics is an important field of study which provides information data on the behavior of molecules within organisms. Knowledge of pharmacokinetic principles helps prescribers adjust dosage more accurately and rapidly. Application of pharmacokinetic principles to individualize pharmacotherapy is termed therapeutic drug monitoring. In this video, we will introduce PK and talk specifically about ADME, which is absorption, distribution, metabolism, and excretion, and the PK compartmental modeling. The information presented in this video should not be used for diagnosing or treating a health problem. Consult your healthcare provider before making any changes to your health plan. Remember to subscribe to this channel so you can be notified of more pharmacy and medicine related videos. Sometimes referred to as PK, pharmacokinetics is a branch of pharmacology that is dedicated to determining the fate of substances administered to a living organism. Chemical xenobiotics, or exogenous products such as drugs, food additives, pesticides, and cosmetics are all investigated by PK studies. It's not just how a molecule acts within the organism that determines its fate from initial administration to complete elimination. However, the organism itself acts upon an introduced chemical substance due to a number of factors. Pharmacokinetics is the study of how the organism affects the substance compared to how the molecule affects the organism. And that's pharmacodynamics. So pharmacodynamics is the study of how the molecule affects the organism. Both PK and PD, or pharmacodynamics, are combined in PK-PD modeling, which influence the dosing, benefit, and adverse effects of administered substances. There are several different pharmacokinetic models used which try to understand how a molecule behaves in and affects the organism. The multi-compartmental model is a commonly used mathematical standard, which we'll talk about. First, let's discuss some very important key terms. Dose is the amount of drug administered, and what I have in italics here is the symbol that you'll see in literature or mathematical equations to highlight what the term is. So dose is identified by D. Dosing interval is the time interval between dose administrations. Maximum serum concentration, or Cmax, is the peak plasma concentration of a drug. Minimum time for Cmax is called Tmax, or the time that's taken to reach the Cmax. Minimum plasma concentration is the lowest concentration that a drug reaches before the next dose is administered. Average plasma concentration is the average plasma concentration of a drug over the dosing interval in steady state. Volume of distribution is the apparent volume in which the drug is distributed. For example, the parameter relating drug concentration in plasma to drug amount in the body. Concentration, as identified as C0 or C steady state, is the amount of drug in a given volume of plasma. Area under the curve is the integral of the concentration time curve, and that could be after any single dose or in steady state, as indicated by the SS in the symbol. Bioavailability, as symbolized by F, is the systemically available fraction of a drug. Absorption half-life is the time required for 50% of a given dose of drug to be absorbed into the systemic circulation. Absorption rate constant is the rate at which a drug enters into the body for oral and other extravascular routes, so does not include intravascular. Elimination half-life is the time required for the concentration of the drug to reach half of its original value. So I do have a separate video on drug half-life that you can refer to. Elimination rate constant is the rate at which a drug is removed from the body. Infusion rate is the rate of infusion required to balance elimination. Clearance is the volume of plasma cleared of the drug per unit time, and fluctuation is the peak-trough fluctuation within one dosing interval at steady state. Okay, so let's now talk about ADME. First, absorption. Absorption is the process that brings a drug from the administration, for example, tablet or capsule, into the systemic circulation. Absorption affects the speed and concentration at which a drug may arrive at its desired location of effect, for example, the plasma. There are many possible methods of drug administration, including but not limited to oral, intravenous, intramuscular, intrathecal, subcutaneous, buccal, rectal, vaginal, oral, ocular, otic, inhaled, nebulized, and transdermal. Each administration method has its own absorption characteristics, advantages, and disadvantages. The absorption process also often includes liberation, or the process by which the drug is released from its pharmaceutical dosage form. This is especially important in the case of oral medications. For instance, an oral medication may be delayed in the throat or esophagus for hours after being taken, delaying the onset of effects or even causing mucosal damage. Often in the stomach, the low pH may begin to chemically react with these drugs before they even arrive in the systemic circulation. Next in the process is distribution. So distribution describes how a substance is spread throughout the body. This varies based on the biochemical properties of the drug as well as the physiology of the individual taking that medication. In the simplest sense, the distribution may be influenced by two main factors, diffusion and convection. These factors may be influenced by the polarity, size, or binding abilities of the drug, the fluid status of the patient, for example, hydration and protein concentrations, or the body habitus of the drug and of the individual. The goal of the distribution is to achieve what is known as the effective drug concentration. This is the concentration of the drug at its designed receptor site. To be effective, a medication must reach its designated compartmental destination, described by the volume of distribution, and not be protein bound to be active. Metabolism is the processing of the drug by the body into subsequent compounds. This is often used to convert the drug into more water-soluble substances that will progress to renal clearance, or in the case of prodrug administration, such as the drug codeine, metabolism may be required to convert the drug into its active form. Different strategies of metabolism may occur in multiple areas throughout the body, such as the gastrointestinal tract, skin, plasma, kidneys, or lungs, but the majority of metabolism is through phase 1, which is the cytochrome P450 metabolism, which I do have a separate video for, and phase 2, which is the UDP glucuronyl transferase reactions in the liver. Phase 1 reactions generally transform substances into polar metabolites by oxidation, allowing phase 2 conjugation reactions to occur. Most commonly, these processes inactivate the drug, convert it to a more hydrophilic metabolite, and allow it to be excreted in the urine or bile. Finally, the last step is excretion. And excretion is the process by which the drug is eliminated from the body. The kidneys most commonly conduct excretion, but for certain drugs, it may be via the lungs, skin, or gastrointestinal tract. Medications may be cleared in the kidneys by passive filtration in the glomerulus or secretion in the tubules, complicated by reabsorption in some compounds. Now let's switch gears to PK modeling, or compartmental modeling analysis. PK models have been developed to simplify conceptualization of the many processes that take place in the interaction between an organism and a chemical substance. Pharmacokinetic modeling may be performed either by non-compartmental or compartmental methods. Multi-compartmental models provide the best approximations to reality. However, the complexity involved in adding parameters with that modeling approach means that monocompartmental models and above all two compartmental models are the most frequently used. The model outputs for a drug can be used in industry, for example, in calculating bioequivalence when designing generic drugs, or in the clinical application of pharmacokinetic concepts. Clinical pharmacokinetics provide many performance guidelines for effective and efficient use of drugs for human health professionals and veterinary medicine. Models generally take the form of mathematical formulas that have a corresponding graphical representation. The use of these models allows an understanding of the characteristics of a molecule, as well as how a particular drug will behave, given information regarding some of its basic characteristics, such as its acid dissociation constant, or pKa, bioavailability and solubility, absorption capacity, and distribution in the organism. A variety of analysis techniques may be used to develop models such as nonlinear regression or curve stripping. In the table on the right side, there is a brief comparison of the compartment and non-compartmental models. For example, the compartment models require elaborate assumptions when non-compartment models don't. For compartment models, curve fitting of experimental data using computers. It is a tedious method, but for non-compartmental, simple algebraic equations are usually sufficient. Compartmental can be used or can be applicable to linear and nonlinear pKs, and non-compartmental is only applicable to linear pK. Compartment models, the C1 time profile is regarded as expression of exponents, while in non-compartmental it's regarded as statistical distribution. For compartment models, these are useful for most of the situations, though assumptions are involved, as we said. For non-compartmental, particularly useful for the applications of clinical PK, bioavailability, and bioequivalence studies. Okay, a little more on non-compartmental analysis. Non-compartmental analysis models are model independent, meaning they do not rely upon assumptions about body compartments, and they tend to provide more consistency. In addition, NCA relies almost exclusively upon algebraic equations to estimate PK parameters, making the analysis less complex than compartmental models. NCAs often prove faster and more cost efficient to conduct, especially when compared to more complex compartmental analyses. For example, compartmental models that are applied to population PK analyses and that rely upon sparse sampling techniques. Total drug exposure is most often estimated by area under the curve methods. Beyond area under the curve exposure measures, parameters such as Cmax, Tmax, clearance, and volume of distribution can also be reported using NCA methods. But beyond that, you really can't use the non-compartmental analysis. NCAs are typically favored for characterizing PK with a single study, including both final analyses and any interim analyses used to make dose escalation decisions. NCAs are the most commonly used approach for establishing the initial exposure characteristics of a drug prior to entry into the clinic. While one compartment model, which we'll talk about as part of the compartmental analysis in the next slide, one compartmental model and non-compartmental analysis both assume that the entirety of the body exists as one container. The difference between the two is that since the one compartmental model is a model-based method, parameters such as clearance can be changed to explore the effects of specific disease states, such as renal impairment or induction of metabolism on the PK characteristics of a drug. Non-compartmental analysis is a model-independent method. where PK parameters are estimated directly from observed data requiring no historical knowledge of the PK characteristics of the drug in the body, for example, absorption rate or rate of elimination. Compartmental modeling is a model-based method used for estimating PK parameters. To apply this method, the body is divided up into hypothetical compartments. Often, the containers used in compartmental modeling do not represent actual physiological tissues in the body, but are used as a proxy so that the PK parameters can be determined. For example, in compartmental modeling, the rates of absorption and/or clearance can be modified between compartments to model the effect of such changes, representing a disease state or induction of metabolism. Compartmental modeling is a general term that refers to a model with at least one compartment. Typical compartmental models have between 1 and 3 compartments, but more compartments can be added to the model depending on the application. For example, in a type of compartmental modeling known as whole body physiologically based pharmacokinetic modeling, or PK, there can be as many as one compartment for each organ of the body. The advantage of compartmental over non-compartmental analysis is the ability to modify parameters and to extrapolate to novel situations. The disadvantage is the difficulty in developing and validating the proper model. Although compartmental models have the potential to realistically model the situation within an organism, models inevitably make simplifying assumptions and will not be applicable in all situations. However complicated and precise a model may be, it still does not truly represent reality despite the effort involved in obtaining various distribution values for a drug. This is because the concept of a distribution volume is a relative concept that is not a true reflection of reality. The choice of model therefore comes down to deciding which one offers the lowest margin of error for the drug involved. Compartmental modeling methods consider the body to consist of a finite number of interconnected, well-mixed, and kinetically homogeneous compartments, for example, the blood, organs, and other tissues. Based on this view, the pharmacokineticist makes certain assumptions and develops models based upon non-linear regression analysis to describe the PK of the drug. When using a compartmental approach, there is the potential for variability in the output of the analysis because the assumptions used to build a PK model may be somewhat different from one pharmacokineticist to another. Compartmental methods have some key advantages, including characterizing PK across multiple studies, exploring PK variability due to intrinsic factors and extrinsic factors such as age, sex, race, renal impairment, and hepatic impairment, and also informing dosing adjustments based upon these factors. I hope you found this video educational and useful. Please remember to like and share. Thank you.